Hi, I'm Dr. Catherine Sears, an assistant professor at Indiana University Simon Cancer Center, and welcome to my lab. The American Cancer Society and their funding has allowed these students and fellows into my lab to make sure they have an early opportunity to learn about research, to get their hands dirty, and to figure out whether they like this and what they like about it. I have been supported for the last 4 years on an American Cancer Society Mentored Research Scholar Grant, which has continued to let me grow in my research and how DNA repair impacts lung cancer early development. So over here we're going to join Washeen, who is making some cigarette smoke extract for us to test the cell response to this. So right now what we're doing is we're basically producing the cigarette smoke, of course in a contained area so we're not exposed. Now she's just taking off the top. Basically we had it in a vacuum, right? So if you compare that to what it looked like before, that's what's happening. So we look at that, we look at the DNA damage, we look at what it does to cells. So let's go look at some of our cells. These cells have a change that allow us to see whether DNA repair has happened or not. So you can see these are the cells. They're actually what we call adherent cells. They're adherent on the plate. These cells kind of grow in a group, but they don't all grow that way. Different cancer cells and different cells in our body grow differently. And so we look at this to see what our cells look like under the microscope, and then we can look at different modifications in different ways. We could treat cells like this with that cigarette smoke that we made and look at their response, whether they're dying, whether they're still alive, why they're dying. That's what we do. Well, thanks for coming to visit my lab today, and thank you all so much for the support you give to cancer research. It's meant a lot in my career development, and I can't wait to see where lung cancer research goes in the future.